Hey everybody, today we are talking about sensory receptors, and then we're going to touch base on touch. Somatosensory receptors are types of afferent receptors, meaning that they collect information and then send it upwards to the central nervous system, primarily the brain. Depending upon which sense we're talking about, the sensory receptors are going to be specialized for that sense and then stimulated by different stimuli. So in this picture here, we have a variety of stimuli and then a variety of receptor types. So we'll talk today about these touch types of receptors that are in connective tissues. We'll talk about free nerve endings. And these work in a similar way to other special senses that you may have learned about in the past. Like, for example, we have photoreceptors that are stimulated by the presence of light waves. In this picture, we see one of the kinocilia receptors that are in the inner ear that are stimulated by movement of the statoconia upon these cilia. So the stimulus is going to be dependent upon what the receptor type is. So we'll talk about touch, pain, cold, heat, and we won't touch today on sound, light, and balance. So the classes of receptors we'll talk about today are going to fall into just 3 different categories of the general category of somatosensory receptors. We'll talk about mechanoreceptors, which are stimulated by mechanical pressure, like tactile or position. Thermoreceptors, which are responsible for temperature, and then chemoreceptors. Chemo is chemical, so those are receptors that are stimulated by the presence of certain chemical substances that give us the perception of pain. I want you to notice in this picture we have an epidermis and we have a dermis, and we have a variety of different receptor types at different levels of the tissue. But if you follow the various receptors, it doesn't really matter what they're sensing. They do share the same general nerve, which means that these things are going to have some overlap. So sometimes pain and cold can have some overlap. Or maybe touch and heat. This is the way a lot of different therapeutics work to get you to ignore the pain sensations you might be feeling. All right, first up are the tactile receptors of the skin. So when we say tactile, we're referring to touch. So we have direct touch receptors that let you know if something is moving across the surface of the skin, if there's persistent pressure on the skin, and it can be very detailed or specific. That will differ between skin with hair or skin without hair. We have pressure receptors, which are embedded deeper in the tissue, and then we have vibratory sensing receptors. The main types of receptors that you should be familiar with are illustrated in an image here, where you can see that depending upon their job, they're located at various places in the tissue. Some of them are very deep within the dermis, and some of them are very superficial at the epidermal layers. This is a direct insight to what their physiology is. If they're deep in the tissue, we're looking at deeper types of stimuli. Some tactile receptors are outlined here, and we also see an accompanying picture. And one thing to keep in mind is not only knowing what the names of these are, what their job is, and then that'll also let you know where they are distributed in the tissue. Another thing to keep in mind is whether or not that skin is glabrous or non-glabrous. So glabrous skin lacks hair, and that's places like your lips, like the soles of your feet, like the genitals, and like the palms of the hand. So various receptor subtypes occur in one or the other, some of them in both, but some are only associated with hair or non-glabrous skin. So first off, we've got free nerve endings. Those are all over the place in your body, and those sense things like touch and pressure. They're extremely superficial, and they let you know literally if something is touching you, whether or not you're wearing clothes, is an example of when these are stimulated. Right next to it is just an expanded tip receptor, which are similar to free nerve endings, but they are just enhanced oscillatory types of receptors on the tips of your fingers in places that are highly sensitive. We have Meissner's corpuscles. Those are located over non-glabrous parts of the skin, and they detect movement. So whether when you're pulling a shirt on, it will disturb the hairs, and that will stimulate some receptors, and then also the Meissner's corpuscle. What co-occurs with Meissner's corpuscles are Merkel's discs. Those tell you about continuous objects. So Meissner's is movement of objects. Merkel's is continuous touch of objects. They are just a specialized type of free nerve ending. They co-occur with Meissner's corpuscles in this place called the egodome receptor. And finally, we have the Krauss's corpuscle. Those detect light, touch, and mechanical vibration. So they have a special name, which is vibrotactile. So vibration and touch. Most of these are in the genitalia only, and that's glabrous, non-hairy tissue, and the highest concentration is within the clitoris. Finishing up our different receptor types, we've got the tactile hair end organ. If we take a look at this, around the base of every hair, we have a free nerve ending that is wrapped around the base. That lets you know whether or not the hair is moving. So that's stimulated by the movement of hair. So like we had the receptors that sensed movement of objects, the tactile hair are a type of receptor that let us know whether or not the hair is moving, which also lets you know if there's movement. We have my favorite ones, the Ruffini's endings. They are easy to remember for me because I like to say rough. Enie's endings, because they're prolonged heavy pressure. Those are the types of receptors that are stimulated by something like a massage where you're getting nice deep pressure. Those are also stimulated by something like sitting in a chair. You're getting deep deformation of the tissues in that case. Finally, we have the Pacinian corpuscles. Those are right here. Those are your tissue vibration. Those are a different vibratory oscillation than the Krauss end bulbs. And they're just located in different places. They're beneath the skin and in the deeper tissues. These two, if you go to some of my neuro lectures, you'll see me talk about those there. There's the Golgi tendon and the muscle spindle, which sense things like tendon pressure and muscle stretch, which we're not going to go through right now. All right, in addition to the tactile receptors, interspersed between them, we also have pain receptors. And those co-occur with things for temperature, which is interesting to remember from a physiologic point of view because heat and cold eventually will be perceived as painful. So you can leave something hot on your hand for a while, but eventually it might start to hurt or become uncomfortable. Same thing with freezing water. If you stick your hand into a cooler, it doesn't feel that bad initially, but eventually it becomes a painful stimuli. So there's a lot of overlap between temperature and pain. But we can see in this picture that the free nerve endings that we spoke about for just touch in the superficial areas of the skin are also really important for things like pain. So they're very superficial and they're easy to stimulate. They're widespread in superficial layers, and some of the internal tissues that are labeled here do possess them as well. But generally the deep tissues, especially the hollow organs of the body, don't have a lot of pain receptors there. So only do you see lots of widespread damage start to lead to pain in those areas. But generally pain is a stimulus that lets you know that something is disrupting your physiology and you need to move away from what that is or correct whatever stimulus is causing that response. And just a note about these different vibratory receptors. So we had tactile, we had tacto-vibratory, and then just vibratory receptors. So Pacinian and the Krauss ones, those do detect different types of vibration. So vibration is just a rapidly repetitive type of mechanical stimulation. The Pacinian corpuscles in the deep tissues actually can pick up really high frequency vibrations, where the Meissner's corpuscles pick up lower frequency vibrations. The genitals, those are supplied by the Krauss end bulbs. All of these vibratory things are going to travel up the same neurologic pathway that all of the somatosensory receptors take. So it's a nice thing to be able to test whether or not a nerve tree is working. So if somebody has neuropathy, we can trace where that damage may have occurred by using simple non-invasive tests like a tuning fork. Another thing I want to mention as well is, in addition to having those touch receptors, those pain and temperature vibratory receptors, there's also an overlap of what we call the tickle and itch system. So the same nerves that supply all of those various receptor types also are stimulated by the presence of something that tickles you. So the mechanoreceptive free nerve endings also in various parts of the body are highly sensitive, but also rapidly adapting, meaning that let's say you're putting on a shirt. You have a really strong stimulus that the shirt is on, but over time, that stimulus kind of dampens down. You don't need that large signal that you're wearing a shirt going on all day. However, when there are tiny little vibratory tactile movements in the superficial layers, that can cause a tickle or an itch response. This is where, if you've ever felt a bug, a tick, or a flea land on your skin, and your first impulse is to flick it away or itch it away, this is a response that travels mainly via C fibers that lets you know that something is on your skin that you need to get rid of. That is one of the jobs that hair has also in the body. If something lands in a hair, that's going to stimulate those hair end organs. We'll feel it moving, and then we'll feel that tickle and itch sensation. But there's a lot of overlap with tickle and itch and the other pain receptors, which we can target with things like opiates, because those, again, that's pain. It's a different type of pain. But what you can start to kind of add together here is, when you think of somebody on opiates, one of the side effects is feeling really itchy. Well, why is that? Because it's affecting this pathway, and this is also something that transmits tickle and itch. And if we're affecting it, we're going to affect more things than just pain, because things are so interspersed. All right, so I mentioned that tickle and itch, they travel by C nerve pathways. Well, what does that even mean? Just a quick note that when we're thinking about nerves, which are just neurons that travel in the periphery of the nervous system, they are classified by how thick they are. And the thicker a neuron is, the more myelin it has on it. So if you look at this picture here, the red in the images, those are just the nerve fibers, and then the yellow on the outside is the myelin. That's what insulates the neuron. That's what allows things to travel more quickly. So the thicker a neuron is, then the more myelin it has, and the better the signal and the faster the signal. Things that are really important that need to alert the body to where it is in space and keep you upright and alive travel via these class A pathways. A lot of things, though, like what we're talking about today, pain, temperature, they travel via these class C pathways that are non-myelinated tissue, and things travel very slowly. This is a very old evolutionary-wise pathway, and this is one of the things that picks up all of those different stimuli we just mentioned. This is also a system in which there's a lot of overlap, so we can get multiple sensations by messing with the system. within this system. So we have these class C fibers, we have class A fibers. You know what? It doesn't really matter which class they are when we think about the nervous pathway. There is a pathway that sensory receptors take, all of these somatosensory receptors we talked about today: touch, pain, temperature, vibration, all of these go up this pathway called the dorsal column medial lemniscal system. This tells you where things travel. They travel in the dorsum columns. They are medial in the spinal column, and they travel through the medial lemniscus. If you think about the knee with the medial meniscus, we have a medial lemniscus up in the cord. So all of these senses will be first stimulated by whatever stimuli makes that receptor fire. So either it's a vibration or it's a tactile response that triggers the mechanoreceptor to fire. That mechanoreceptor in the skin is going to fire, meaning it has an action potential. That response is going to start traveling through that nerve. So if this is on the tip of your finger, it will go from the tip of your finger all the way up to a level of the spinal cord. It's going to travel through the dorsal roots, which is where sensory information travels through, and it will stay on the same side of the spinal cord and travel all the way up uninterrupted until it gets to the medulla. Once we get to the level of the medulla, we see that the second neuron in line, what we call the second order neuron, picks up and synapses with the first order neuron right here, and then decussates or skips over to the other side and then travels again uninterrupted up to the thalamus. The thalamus is the relay station for all sensory information. It's called thalamic relay. All sensory information should synapse here. So this is no different. We synapse with the third order neuron, so the third in order from the initial stimuli, to travel to the part of the brain that needs to tell us what this area or what that stimulus is, so it makes sense of that information. That area is the somatosensory cortex. So this is a simple spinal pathway. The dorsal column medial lemniscal system, receptor to spinal nerve up to medulla, second order neuron synapses there, decussates over at the medial lemniscus, travels up to the thalamus, and then synapses with the third order neuron, which goes from the thalamus to the somatosensory cortex. And that's the way touch travels in our body up to our brain so that we can make sense of whatever it was. Up in the somatosensory cortex, we have a really interesting thing called the sensory homunculus, this little man, this little brain. So essentially, we can map the entire surface of the cortex with the area of the body that those nerves originate from. Things that are heavily innervated are overrepresented by just a dramatic exaggerated size of that here. So if we look at this part of the cortex, most of this face is dedicated to an area of the brain that tells you stimulation from the upper lip, the lips, the lower lip, and then some from the face. But if we look at things like, your wrist and forearm, we have very low stimulation pathways coming from that. So there's definitely areas in your body that are very sensitive, which are very innervated. Areas of your body that are not that sensitive, they're lowly innervated. If we take a zoom out and look at it from the cortical aspect of the brain, this is the area we're talking about, the somatosensory cortex. There's two different areas. There are areas in the brain called the Brodmann's areas. Don't need to worry about them much today, but we are categorizing these by just somatosensory area 1 and 2. 1 is this area abutting the primary motor cortex, full of all of these innervated areas, and somatosensory area 2 is right here behind that. Those are going to then collect information from those sensory receptors in the body so that it can send that information to the primary motor cortex, which is the area in your brain that allows you to make muscle movements. So if you get a stimulation that something is on your arm and it's buzzing and making a vibration, then that information can go right here to stimulate you to move your hand and to brush the thing off that's causing that. When we really have a nice strong stimulus from one of these mechanical receptors, then there is a lot of cortical involvement and lots of neuronal involvement. We're relatively good at getting single point discrimination, meaning if I touch you on your palm and then ask you where I touched you, you're pretty good at pointing to where that is. Where things get complicated is when you introduce a second point. So if we were to discriminate between 2 points of contact, like if I put 2 of my fingers on your back, maybe at your scapula, and then move those fingers closer and closer together and asked you to let me know when my fingers were completely together, we're not that good at doing it. We actually estimate that things are touching in 2-point discrimination pretty far apart. So my point is that we're pretty good at single point discrimination. We're very poor at 2-point discrimination. However, all of this overlap in the system allows us to take advantage of the fact that we have multiple things from many different input coming in and taking the same pathway. So we can utilize different types of stimuli to overrun an initial message. That's why something like putting on a really hot topical like capsaicin cream to the surface of the skin might make your skin feel really hot. But also you would use that because initially you would have pain there. So we can distract our brain from one stimuli by just using another stimuli. We have so much overlap in this pathway, and that should start to unlock some of the doorways to you into how pharmacotherapeutics work. So that's what I want you to know about touch and vibration, where the receptors are, what pathway they travel in, and what they attend to. That's all for today. I hope you learned something. Everybody stay smart.